Hello, and thank you for joining me today as we wrap up our introductory unit to A and P. Today we're going to talk about cell organelles, we're going to talk about transport and different types of transport, and we're going to talk about mitosis. So at the end of this lesson, you're going to be able to name and provide the function for at least 5 organelles in the body. We're going to talk about differences between diffusion, facilitated diffusion, and osmosis. We will be able to explain differences between hypertonic, hypotonic, and isotonic solutions in regards to how cells react to them. We'll be able to use the terms endocytosis and exocytosis when you describe the movement of materials across the cell membrane. We're going to look at 2 forms of endocytosis and you'll be able to differentiate between them. And I want to know what happens in each phase of the cell cycle. So here we go. To start with, we have organelles in the body. There's a number of them, including things like your nucleus, which stores your genetic information. Within that, you have your nucleoplasm, which is the fluid. You have your nucleolus, which is in the center. Your envelope is the covering, and all of that stores DNA. Your cytoplasm is the fluid that fills your cell. Cytoplasm is actually the space between the membrane and the nucleus. Cytosol is the fluid, but it's all the same. It works for this class. Your endoplasmic reticulum produces and processes or distributes proteins and lipids throughout the cell, moves things across the cell. Your ribosomes produce proteins, are used to make proteins. Your Golgi complex, I call it a Golgi apparatus, acts like the UPS of your cell, processes and delivers different proteins throughout the cell and sends them to other cells and gets them where they need to go. And we have your mitochondria. This is your powerhouse. Performs cellular respiration, produces energy, allows you to function and do the things that you like to do. More organelles. Vacuoles are a fluid-filled sac used to store materials, specifically water. Vesicles are transport packaging. So your Golgi apparatus surrounds the material to be transported in a vesicle, which then moves it throughout the cell. Lysosomes digest material. So lyso means to split. So a lysosome splits things apart, breaks things up, destroys things that aren't needed. Centriole is a spindle used in mitosis. Your microtubule, also found in mitosis, structurally supports the cell, aids it in mitosis. Your cell membrane protects and encompasses everything, keeps it all together. And we have microvilli, which allows for more surface area, which is important in things like the digestive system. So reviewing organelles. Which organelle contains the instructions for everything, keeps you alive, allows you to function and process and fix things and build and repair and maintain. Which organelle is responsible for preparing materials to be sent out of the cell? So what's our shipping center known as? Name 5 organelles. Not these 2. Name 5 different organelles and what they do for the body. Once you've done that, we'll continue on. All right, we're talking cell transport. Your cells are surrounded by what we call the cell membrane, which is our covering. This covering determines what can enter and what can exit the cell. That's the important piece. And so this cell membrane allows certain things to pass through but keeps others out. And there are a couple ways that we can get things into and out of the cell. The first of those is called diffusion. Diffusion is the tendency for atoms, molecules, ions, something in a fluid to travel down the concentration gradient. So from high concentrations to low concentrations. So diffusion goes from high to low to establish this equilibrium. When there is no high and low, it is equal on either side. That is diffusion. Diffusion happens without any energy transport. It's a passive process, so meaning things happen naturally. It normally occurs. Sometimes we have substances that are too big to go from one side of our barrier to the other, our cell membrane to the other. In this case, we have what's called facilitated diffusion. This is where you have a transport molecule or transport protein. So if I have a cell and I'm trying to get this particle into the cell, but it's too big to pass through the membrane, the cell will put a protein channel here that this molecule can then pass through to get into the cell. And because there's more of these molecules out here than in the cell, it'll pass through normally because that's diffusion taking place. So that is diffusion. The only difference between this and facilitated diffusion and regular diffusion is this transport molecule or transport protein, carrier protein, that's found in the membrane. Then we have osmosis. Osmosis is specifically the diffusion of water molecules. So most things we talk about diffuse in water. For example, if I have a beaker that's got water on this side and it has salt water on this side, this is a semi-permeable membrane. This is like your cell membrane being in here that doesn't allow anything to just pass through. So in this case, if my salt is too big to go across, it can't go across there, it's too big, this water will actually go across this way. And so if you missed this lesson, please talk to me. I will show you a video showing you kind of what happens in osmosis and why this works. So with movement, we have different terms for solutions that we use. These three terms are called hypertonic, hypotonic, and we have isotonic we'll talk about on the next one. Next slide. In a hypertonic solution, think of students that are really hyper. If you're in a classroom and you're hyper, what do you want to do? You want to get out of that classroom, right? You want to be in a larger space. And so what happens in a hypertonic solution, I have water inside my cell, and I have a bunch of solute particles outside my cell. So for example, salt water. If there's more salt outside of the cell than inside of the cell, the water is going to leave the cell and go to the bigger area. When this happens, your cell is going to shrivel up and die. So more water leaves the cell than enters the cell, so your cell shrivels up and dies. In a hypotonic solution, for hypotonic, think of, if I called it a hypotonic solution, hippos are very large. So in this case, I have a cell that has some salt in it. And I put it in pure water that doesn't have salt. This water is going to rush into the cell to try to dilute it out. So it's the same on the outside as the inside. So because the water rushes in, the cell gets big. It becomes a hippo over here. Your cells enlarge. If too much water rushes in, your cell can burst and become non-functional. And so filtration is how we can kind of look at these different substances. So this is using a membrane to separate substances, usually your larger solids from your smaller particles. And this is where we see your hypotonic, your hypotonic and your hypertonic solutions, or isotonic. And then, so I guess an isotonic solution has water enters and leaves the cell at the same rate. So the cell stays the same size. So in an isotonic solution, water enters and leaves the cell at the same rate, so the cells can continue to function as they are. We talked about diffusion. Diffusion is a passive process that doesn't require energy. Some molecules are too big to pass through without any energy, or they're moving from low concentrations to high concentrations. In this case, we have what's called active transport. So this moves particles up the concentration gradient, so from low to high. This requires energy, hence the term active. Roughly 40% of a cell's energy can be used for this process to get it the things it needs. We have two broad categories of active transport. One is endocytosis, one is exocytosis. In endocytosis, we're looking at particles coming endo or into the cell. So endocytosis goes into the cell. Exocytosis exits So endocytosis is into, exocytosis exits. So it's the reverse process. Substances are exited or excreted through vesicles. With endocytosis, bringing things in, we have two main types. One is called Pinocytosis, which moves liquids. This is like cell drinking. We also have Phagocytosis, which is like cell eating. It brings in solids. So your white blood cells perform phagocytosis to destroy cells that aren't supposed to be there. Then we also have this receptor-mediated endocytosis, which is just bringing in the specific things that are supposed to be. So receptor-mediated means this is being controlled. It's got to match with the receptor on the outside in order to be taken into the cell. Molecules binding to the receptor, they're taken into the cell, then they can be used. All right. How are diffusion, osmosis, and facilitated diffusion different? These three terms. How are isotonic, hypotonic, and hypertonic solutions different? What happens to a cell if it's placed in each of those? What are my two main forms of active transport? And how are pinocytosis and phagocytosis different? Last thing we're going to talk about after you answer this is mitosis, and then we'll be done with today's lesson. So interphase is when, so the cell cycle is what we're going to talk about. Interphase is when your cell does what it's supposed to do. Your cells will spend most of their time in interphase. At the end of this, your cell components duplicate, which allows us to perform mitosis. So in this process, our cellular components divide and replicate. In mitosis, the cell divides. And so interphase itself can be split into three main parts. We have G1, S, and G2. G1 is growth 1, S is synthesis, and G2 is growth 2 or gap 2. So G1 is the first growth phase. This is when everything in your cell is duplicated except for DNA. So you double your mitochondria. You duplicate your ribosomes. You duplicate your endoplasmic reticulum. You make extra so your cell can divide. In S phase, synthesis, you are synthesizing your DNA. And then in G2, this is kind of your second check, your double check, your final check to make sure you have everything you need to divide. If not, your cell will backtrack and fix things. Then your cell performs what's called mitosis. This is cell division in somatic cells, so non-sperm and egg cells. So it performs genetically identical cells, has all 46 chromosomes. There are four main phases. I think of it as PMAT. Prophase, metaphase, anaphase, and telophase. In prophase, your nuclear membrane breaks down. Your DNA forms into coils, wraps itself up. It's bundled there. Your spindle fibers attach to your centromeres, and everything comes together. In metaphase, your DNA meets in the middle in metaphase, all M's. So meet in the middle in metaphase is what happens here. DNA lines up. In anaphase, after we meet in the middle, we have to pull them apart, so that's what happens there. And then in telophase, your membrane pinches off and forms two separate cells, which is what we get here. Two identical separate cells. All right, that is today's lesson. Make sure you can answer these questions. What are the three parts of interphase? What happens in each? What are the four steps of mitosis? What happens in each? And what would happen if we lost control of the cell cycle and cells continue to divide? Take a look at those. Make sure you can answer them. If you have questions, please come see me. I look forward to discussing with you spending the rest of our time talking about anatomy with you as the year progresses.